G'day everyone, I'm Jake Phillips, Breed Development Officer with Angus Australia, and welcome back to the Stock Agents Toolkit. In this module of the Stock Agents Toolkit, we're going to be going in more than skin deep. That's right, it's all about DNA. And you're probably wondering, well, yes, I know cattle have got DNA, but did you know that we routinely use an animal's DNA for three main reasons? And they are to determine who the sire and dam are, which is otherwise called parent verification, to conduct a genomic test, which is often used on young animals to predict their genetic merit for certain traits that particularly may not be expressed until much later in their life, and also to establish if the animal is carrying any recessive genes for genetic disorders. All of this information will be available in Angus sale catalogues or in Angus database search results when looking up a certain individual. Angus breeders in Australia have been submitting tens of thousands of DNA samples on animals each year, which is a clear commitment to the science and the process that is helping to make Angus breeders some of the most progressive and innovative breeders anywhere in the world. And this is because they're utilising DNA technology in their quest for genetic gain. Currently, DNA is submitted by Angus breeders in two main ways, the first of which is hair collection and the second is a tissue sample. The use of DNA technology in the seed stock sector has given a much needed boost to the power and accuracy of the genetic selection, particularly in traits that are difficult to measure, or for breeders seeking more accuracy on traits earlier in an animal's life. So let's get started by taking a deep dive into the process of parent verification. In general, parentage verification is based upon the detection of genes inherited and present in a calf's DNA. Progeny receive one chromosome from each of its parents, and so genetic markers are passed down from parent to offspring. Parentage verification is based on exclusion, proving that the animal is not the offspring of a sire or dam, rather than proving that it is. What this means is that, for example, if a marker is present in a calf but not in the nominated parents, then the calf is excluded as being a possible offspring of that particular mating. Given the level of accuracy this process has, more than 99% of incorrect matings will be detected by DNA parentage analysis. The reason it is important to consider this when looking at bulls with your client is that bulls that have been parent verified are representing the pedigree and the associated genetic performance assumptions accurately. And this is because they have a quality assurance step in place. DNA parentage verification is being used by many proactive Angus breeders as a level of quality assurance over the parentage claims, which also aids in ironing out any discrepancies. Angus Australia also have two optional programs that appear in some catalogues. If a catalogue has all been verified for the sire, a sire assured logo and description will be associated with the catalogue. If a catalogue has all been verified for both sire and dam, a parentage assured logo and description will be associated with that catalogue. Parent verification is rapidly becoming a tool that is helping many breeders to conduct complex and intensive genetic improvement programs by offering a unique tool to assist with pedigree recording and trait prediction on hard to measure traits. Let's have a look at the next use of DNA technology, which is genomics. Genomics is the study of an entire set of genes found in living things. A genome is an organism's complete set of DNA. All of the information needed to build that organism is contained in their DNA. The main difference between genomics and genetics is that genetics may look at a single gene, but genomics looks at all genes and how they interact to influence growth and development of an organism, or in this case, an Angus animal. Genomics gives cattle breeders more confidence and more accuracy on genetic merit far in advance to when it could otherwise be measured, particularly in conventional and traditional methods. Genomic testing can help predict future profitability by potentially indicating the likelihood of desirable traits to be carried and expressed in a particular animal. This in turn allows producers to optimize the profitability of their herds by making different or alternative animal selection and strategic breeding decisions much earlier than they would otherwise. The reason genomics has become a go-to tool for many Angus breeders is that traits such as the colour of coat or being polled or horned are generally controlled by one gene. However, other traits important to economic production of beef, such as calving ease, fertility traits, traits important to carcass value, growth traits, feed efficiency traits, and even docility and structural traits are much more complex and controlled by multi-trait combinations. Importantly, a raw genomics result is then incorporated into the routine TACE analysis conducted by Angus Australia each fortnight. This includes the pedigree, the performance, and the genomic information. This results in EBVs being generated and being able to be utilised for breeding and selection decisions. So in summary, genomics gives higher accuracy on EBVs on younger animals. It gives more comparison between animals that aren't necessarily closely linked in pedigree, but they share the same gene markers, and gives genetic merit and higher accuracy in hard-to-measure traits. The final core usage of DNA technology is for testing of recessive genetic conditions. A genetic condition is caused by an abnormality in an individual's DNA. These abnormalities can range from a single gene mutation to the addition or loss of an entire chromosome. Genetic conditions have been identified in most species, including humans, and over 400 genetic conditions have been identified in beef cattle. Approximately 1/4 of these are caused by a single gene mutation, making them reasonably easy to manage through DNA testing. Historically, before DNA tests were available, genetic conditions were managed by extensive progeny testing, or by eradicating all known relatives of the affected animal. This resulted in widespread production losses and the potential loss of superior genetics. Angus Australia currently monitors 9 genetic conditions. Here is a list of the genetic conditions that we currently monitor. Genetic conditions have different modes of inheritance. All genetic conditions monitored by Angus Australia follow a single recessive gene mode of inheritance, making them easy to manage. These single gene recessive genetic conditions result in 3 possible genotypes. The first of which is an animal will test free of that genetic condition, meaning that the animal is normal and carries 2 copies of the normal form and no copies of the mutation. The second outcome is that the animal is in fact a carrier, meaning the animal may look normal but carries 1 copy of the mutation, which can then be passed on to their progeny. And the third outcome is that the animal is actually affected and will likely present with physical signs of the condition. This diagram provides an illustration of the possible mating outcomes from different mating combinations for a single gene recessive genetic condition. Angus Australia currently reports the genetic condition status of animals within Angus Tech. You will also find these results in sale catalogues listed next to the animal registration details, and you will also observe results being presented as free, carrier, free untested, or possibly free untested with a percentage sign. These results indicate the chance the individual is a carrier of the recessive gene based on pedigree, but hasn't yet been tested. Angus seedstock breeders are proactive in monitoring these genetic conditions, and in the event you or your client notice something of interest in this area, we would encourage you to please talk to the breeder to understand more about what this means for the animal and for the client. Or feel free to give the Breed Development Officer team a call at Angus Australia. In summary, DNA is rapidly becoming an influence on the breeding and selection decision for many Angus breeders across the world. What DNA does is to provide a quality assurance step to the pedigree being recorded for a certain individual, which gives the breeder and the purchaser more confidence in that record. DNA, when used in genomics, allows for higher accuracy EVVs to be achieved, as it monitors and evaluates the pairing of key gene markers for certain traits, particularly in hard-to-measure traits, or in traits that can't be measured effectively until the animal is much older. This is a valuable tool for comparison of animals across herds and even across countries. And finally, the other key piece of DNA technology being used is for monitoring the prevalence of recessive genes that cause production losses. When interpreting information derived from DNA technologies with your client, or when searching through sale catalogues, be assured that these tools are helping to provide more accurate information and can be considered as just one part of the information being presented to you for your appraisal and for inclusion into your selection criteria. Next time on the Stock Agents Toolkit, we're going to be putting all of this theory into practice. That's right, it's now time to define your client's market and their breeding objectives. And part of that will be considering some key traits of importance. But for now, it's time to hit the road. We'll see you again soon as we continue to unpack the Stock Agents Toolkit.